Alright, in this video I'm going to talk about the chemical properties of sugar. So the first thing I want to talk about here is just comparing some of the sugars based on their sweetness percent. And so this is based on sucrose being sort of the standard. That's the 100% sweet. Right here you can see. And so these sugars over here, things like glucose, are less sweet than sucrose. As we'll see, sucrose is a disaccharide of glucose and fructose. Fructose, you see here, is actually quite a bit sweeter than even sucrose, but also glucose. We have here, this is high fructose corn syrup, which is about the same sweetness as sucrose. Then we can see here aspartame, which is actually not a sugar, and we'll look at aspartame later on in this video, but we see that that is extremely sweet. This goes quite a bit beyond even things like high fructose corn syrup or fructose, which is why you only need to add a very small amount of aspartame into a drink to give it the same sweetness that you would if you had sucrose, which is cane sugar. And so that's why aspartame can be a low-calorie sweetener substitute. Uh, then we see down here, we have things like lactose, of lactose intolerance fame, which is found in milk. We see that's not very sweet, which is why milk doesn't taste like, well, it doesn't taste like chocolate milk, which would contain things like sucrose or high fructose corn syrup. And so now let's get into the actual chemical properties of carbohydrates. And so we see over here we have different types of sugars. So these three here are called epimers of each other. Epimer. And they are different in that their hydroxyl groups point in different directions. These carbons that we see that these are attached to are chiral carbons. And so the direction in which their functional groups point, it matters. I'll get into chirality here in just a little bit, but the point is, so in glucose we have this carbon right here is a little bit different, and I'll talk about that in a little bit. That's actually called an anomeric carbon, but these other ones here, number 2, 3, and 4 in particular, are epimeric carbons, and their hydroxyl groups pointing in different directions make them different sugars. So glucose, I always just try to remember, is down, up, down. And so then if we change one of them, so if we change this fourth carbon to up, so it's down, up, up, then we get galactose. If we keep the third and fourth carbon the same, but we change the second carbon, so it's up, up, down, then we get mannose. And so glucose and fructose, and we'll get into this later as well, these are actually what are called Aldose-Ketose isomers, and that's a little bit different. So you can see that fructose is a 5-member ring, where these other ones are 6-member rings, and that will have to do with this aldose-ketose isomerization. There are a few other ways we can alter the sugars. I have just a few of them listed here. So one of them is we can phosphorylate one of these hydroxyl groups. So here we see that glucose using ATP, which gives a phosphate, adenosine triphosphate, turns into adenosine diphosphate by giving off a phosphate, which has donated to this sugar, making it from glucose to glucose 6-phosphate, because this is the 6 carbon here that the phosphate is on. And we'll run into this a lot when we get into sugar metabolism, this phosphorylating of sugars. Another is you can deoxygenate one of the carbons. So the sort of quintessential example of that is going from ribose to deoxyribose. Deoxyribose as in DNA, deoxyribose nucleic acid, which I will cover in more detail in a later video. But we see here that on the second carbon, we're just changing it from a hydroxyl to a hydrogen. So deoxy, it's getting rid of that oxygen. Then we can also have sugars with modifications, such as putting an amine here. And so this is called glucosamine. I'll get into what the D means shortly. So it's glucose, so it has the down, up, down, but one of the hydroxyl groups is changed into a primary amine group. So glucose amine. And if you're into things like nutrition, you might have heard of this in terms of joint health. So glucosamine molecules are used in the synovial fluid between our joints in our body. And so that's a use for something like glucosamine. But I just wanted to point out here that you can make this alteration by changing the hydroxyl into an amine. Alright, so now let's get into this D and L. And that has to do with what I was talking about earlier with the chirality. And chirality just means that there are mirror images of things. So you can see this D glucose and the L glucose. If we had a mirror right here, are exact mirror images of each other. And this is chemically important. What's important in biochemistry is all of the glucose we use in our diet, for instance, is D-glucose, which stands for dextrorotatory. So this is the biologically interesting version of it, where L-glucose is levorotatory. So I'll get into what these terms are sort of indicating here in a moment. But I just wanted to show this mirror image idea. If we put a mirror between, then the molecule is sort of like our hands. We cannot superimpose our hands. No matter how we rotate our right hand, it will never superimpose completely on our left hand. And that's the same with chiral molecules. So if we see that if we took this molecule here and just sort of reoriented it around in space, it will never completely superimpose on this molecule here. In the dextrorotatory and levorotatory has to do with the way that it alters plane polarized light. So we see we have a light source here which is giving off light in every polarization. Then we shine it through a polarizer, which now you see that the light is plane polarized. Its wave is going in just a single direction like this. Then we have it go through our sample, And then what happens is something called Birefringence, and that's the difference in the way that the different molecules interact with light. So it's the difference in the refractory index. And so what happens is one of these molecules will refract the light different, and so that causes it to take on this orientation here. It's this polarization here. Then the other molecule will cause it to take on this polarization here. And then when we analyze it, we combine these polarizations back together, and we see that the constructive interference of this and this, for instance, gives us polarization in this direction, and the other constructive interference in the trough gives us polarization in this direction. And so we see that if one of the molecules is polarizing it in this way, the other one is polarizing it in this way, then if one of them, if this one, for instance, is more prominent, if there's a higher concentration of this, then we're going to have more polarization this way than this way. And so that changes the direction of the entire polarization from where it was after we put it through our polarizer. And so we can see the change in the polarization of the light based on the relative concentrations of these two molecules, which are called enantiomers of each other. So each of these are an enantiomer of each other. When we talk about one, essentially we say that it is chiral. This is a chiral molecule. When we're talking about both sort of relative to each other, then we say that they are enantiomers of each other. And so this light rotation, this polarization, is sort of relative. There's no sort of intrinsic thing in the molecule that we can say is definitely one way or the other. So what we actually use are these what are called the Kahn-Ingold Prelog or CIP Priority Rules. And what that is, thought I had a diagram for that. Guess you'll have to live with my crummy drawings. And so what that means is if we have a chiral molecule, say we have a carbon, and we want to look at it so that the molecule with the lowest priority, which so lone pair is lowest priority, but usually we're talking about hydrogens. So we want to look at it with the hydrogen pointing in the back, and then we can have our R1, so we'll call that R1, R2, R3, and I'm putting 1, 2, 3 as the ranking of the constituent. So we can just say that these are any constituent, but R1 is a lower priority than R2, which is a lower priority than R3. And so what we want to do here with our lowest priority in the back, R1 is higher priority than H, R2 higher priority than R1, R3 higher priority than R2. And then we just go in a counterclockwise circle between them. And this gives us our what's called the R the R chirality, whereas if we had something like this with it going in the counterclockwise direction, so the increase in priority is going counterclockwise, then we are in the S version of it. So we can have an S or an R and an S chirality depending on which way our priority is going here. And so this table is just showing the different priorities uh, that have been given by chemists. And what you can see here is it's mostly based on just so we go from hydrogen and then deuterium, which is just an isotrope of hydrogen. So if we had something that, you know, uh, maybe we have our H there, and then we had a deuterium there, then maybe we had a CH3 here, and then we go down and maybe we have a CH2CH3. Then with this deuterium is this priority, then our CH3, which is methyl, is this priority, then our ethyl is this priority. So we would go around counterclockwise, which would mean this carbon here has an S chirality. And so you can see if you just, as you're adding these groups on in place of our R groups here, then we want to go around in the direction of our increasing priority. That's how we determine our R and S chirality. Okay, so that's the idea of chirality. So let's get into sugars here, and just keep in mind that in our biologically sort of relevant sugars, we are always talking about the D glucose, which is based on the light, the D-L system, the D and L system. There is no correlation with the RS system. The DL and the RS are not correlated. You cannot determine that something is R or S just based on the fact that it is D, which you can see here, for instance. So one of these, if we're looking at this epimer of glucose, going from glucose to galactose, this is changing between R and S, and yet both of these sugars, the glucose and the galactose, are both D sugars. They remain D, even though one of these chiral carbons is changing from R to S. And I guess that's another distinction you can make, is that the R and S has to do with each particular carbon, where the D and L is talking about the entire, well, should include that one too, the entire molecule. So the D and L is the entire molecule. The R and S is looking at each specific chiral carbon. And so, like I said, just keep in mind we are using D sugars in our biologically relevant carbohydrates. And so this is just showing that that our glucose can exist in this linear form here, or in the ring structure. And the thing to notice with the linear form is we have this right here, which is an aldehyde. And so you can actually have sugars, and we'll see this later, where instead of the aldehyde, this double bonded oxygen would actually be on a different carbon, and then we would have a ketone. And sort of suggestively, I talked about the Aldose-Ketose isomers. So an Aldose-Ketose isomer is one where we change the double bonded oxygen from here to here. And that goes from aldose to ketose, aldehyde to ketone. So we'll run into that here in a little bit. So I just want to talk about here when we go from the linear to the the ring structure, we can change. That's why they call this the anomeric carbon. That's why it gets its own special name, is because we can actually have the hydroxyl group going down or going up, and this gives us our either our alpha D glucose. So alpha is with the hydroxyl going down, or in this chair conformation is called axial, and the beta version has it going up, or what's called equatorial. Uh, and so this says that there, well, it doesn't just say, it is the case that there is 30, it's 36% in this form and 64% in this form. Uh, that adds up to 100%. It's not exactly 100% because there is a very small amount that if you take a snapshot at any given time, you will find this, but it will be at less than 1%. But so if we're in equilibrium with this, if it goes to this, then it's either going to quickly snap back into this, or it's going to snap over into this. And if you have your glucose in solution uh, at any, and you take a snapshot of all the molecules of it at any given time, about 64% of them will be in this this beta anomeric form, and around 36% will be in this alpha anomeric form. Less than 1% will be sitting in this linear form. And so the reason that we can have this difference in the ring structure in the anomeric carbon has to do with the way that so this shows this CN group. What's actually acting as the nucleophile in the instance of sugar is actually this oxygen here. So you see this is the fifth carbon right here, and so that's the fifth carbon here, and this is from this hydroxyl group here. And so this hydroxyl group is actually coming and attacking that carbon, or it can go from the other direction up to here and attack the carbon from there. And we can see that here. So if we just kind of pretend that this CN here is actually our hydroxyl group, so going to the rest of our molecule, so this would have a hydrogen on it, but this can attack from either side of the plane of this this carbonyl group here, which in the glucose instance would be an aldehyde. And so we see that that what happens is when we attack from either side, we can end up with these two different chiralities on our carbon. So when it attacks from here, then we end up with this chirality. So on here, where it's the CN, but we'll pretend this is our hydroxyl group, which goes on to be attached to the rest of the molecule, which is it's also attached there because it's a ring. And so we see that we end up with a different chirality based on which direction our nucleophile in the sugar is this hydroxyl group attacks the plane of this carbonyl. So we can see here. So this is just talking about this this Berge-Dunitz angle. So when it attacks the carbonyl, it attacks at this 109-degree angle. That would be the same as if we were attacking from this. This would also be 109. And so you can see that it's coming at the pi orbitals in which is the double bond in our carbon here. So it has to align. This is that orbital steering here that I mentioned in a previous video on bonding theory. So the bond has to form with this p orbital here. And then we can think about it in what's called the Falcon or Falcon on version. And so if we're looking at our carbon sort of head on, so if we have a carbon bonded to a carbon and this has other groups bound to it, and this has other groups bound to it, then maybe H's, which is what we have going on here. If we sort of look at it from this direction, then we would see our first carbon here, and the other carbon is behind it. And then we would have our R1, R2 from this first carbon with the H, which we'll call H1. That's H2. And then we want to see where these other groups in the back, and so we see here that we have the circle going around this carbon, and so what we would see coming from the back is our H2, which I have pointing up, so I have this rotated around this single bond here, and so our R4 would be pointing out that way, our R3 pointing out that way, and we want it to be in a conformation where there is the least amount of steric hindrance between these groups. That's why I've put the H is pointing in these opposite directions so that the steric hindrance is less between these R groups and the group that we would see in the other carbon here. And so this is just looking at that nucleophilic attack on the carbon in the front here, and that would be occurring in this direction, for instance. And this is favored because we're rotated around that single bond in such a way that that we have the least amount of steric hindrance going on. And so this would be the favored way of doing it. So this is something, it's not as relevant for carbohydrates, this Falcon on model, as far as the stereo selection of our alpha and beta. But this idea is something we will run into more in the future. So I wanted to incorporate it in here, where we first started sort of getting deep into this idea of nucleophilic attacks on the carbonyl carbon. What's primarily determining the relative abundance of the alpha glucose and the beta glucose is actually the steric hindrance of the ring structures themselves. So that's what the chair conformation is good at showing, is we see that this hydroxyl here is sort of being crowded out by this hydroxyl here. So the hydroxyl on the anomeric carbon, on the one carbon, is sort of clashing with the hydroxyl group here on the second carbon, and so that disfavors this alpha conformation. Whereas when we are in the equatorial form, then we just have this hydrogen down here, which is much smaller, and so there is less steric hindrance with this hydroxyl on the second carbon here, and so that's why this equatorial, this beta hydroxyl is more favored, and that's why our abundance of it is higher than the alpha glucose. Then here I wanted to sort of touch on this idea of the epimers again. So we saw up here the epimers in the ring structure is the hydroxyl pointing in different directions on our carbon. And we can see that here in the linear form. So we can have the C2 epimers of each other, where on the second carbon we get the change in the direction of the hydroxyl group on the C2 carbon, or C4. Obviously we can have C3 and C5 epimers as well. And so epimerization can occur through this mechanism here, where we end up with this enolate ion. I'm not going to go through the whole thing. We'll talk more about this kind of chemistry in a moment when we are actually talking about the Aldose-Ketose isomerization, which is more relevant for us. But we see that through this mechanism, we have our OH here changing directions on the carbon, which is giving us a C2 epimer. So the C2 epimer of D-glucose is just D-mannose. So we saw that up here. We saw that changing the chirality on our second carbon changed from glucose to mannose. So going back to this idea of the alpha and the beta anomers on our carbons, we can actually get different versions. So when we polymerize our carbohydrates, our sugars, the anomeric carbon is important in the sorts of bonds we form. So here in amylopectin, we have these alpha 1,4 what are called glycosidic bonds, and that is a bond from the alpha anomeric carbon to the fourth carbon on another glucose. So if you remember glucose, we had our hydroxyls down, up, down. So this bond is down on both of these, because we're in the alpha anomeric carbon here. So that's why it's called an alpha 1,4 glycosidic bond. The anomeric carbon is the first carbon, and it's alpha, so alpha 1, to the fourth carbon of the next glucose in the chain, so 4. And glycosidic bond is just the name that has been given to this bond. And we can also have branches coming off, and that's why we have alpha 1, which is here our anomeric carbon, alpha 1. Then the 6th carbon on this glucose, so 6, and that's just another glycosidic bond. And so amylopectin then is starch that contains this these alpha 1,6 glycosidic bonds, where amylose is the version of starch that only has the alpha 1,4 glycosidic bonds. So you can see here the alpha 1 anomeric carbon, the fourth carbon, and this just means that there is a repeat of this bonding going on here. So this could be a very long chain, up to 300 to 600 of these long. So amylopectin and amylose are versions of glucose that you find in starch, and so both of these make up starch. Okay, but now if we go over here and we're instead using beta 1,4 glycosidic bonds, glycosidic glycosidic bonds. So we're using beta on our anomeric carbon instead of alpha. So you can see that here, instead of going down, it's pointing up. Then what we end up having here is, so you can see, first you can see our sugar is changing directions in both of them, because this fourth carbon still has to be pointing down, so we actually have to rotate it upside down in order for this to be occurring. And so it changes sort of the direction of each sugar in the chain here. And then what that does is it allows these hydrogen bonds in the chain, in the actual chain, to be occurring between the hydroxyl on the second carbon and the hydroxyl on the sixth carbon. We have these hydrogen bonds going on, these intra-chain hydrogen bonds, and we also have these interchain hydrogen bonds going on in this version of it, and this is called cellulose, which is a version of polysaccharides, of sugar polymers that humans cannot digest. So this is indigestible to us, and you actually find this a lot in things like the cell walls in plants. Use this structure, which is a more rigid structure because it has these hydrogen bonds going on, which help to make this a more rigid structure. And so that's the importance of the alpha and beta anomeric carbons. So starch is obviously digestible by humans, where if we just change that anomeric carbon from the alpha version to the beta version, and then polymerize it, then we get this cellulose, which is not digestible. Down here, I have the formation of sucrose, which we talked about earlier, which is this. We take glucose and fructose, and we form this alpha-1 beta-2 glycosidic linkage. And so the glucose is in the alpha form, where our anomeric carbon is pointing down, but the fructose it's in the beta anomer, and so we form this bond that's alpha on the glucose and beta on the fructose, and we form this alpha-1 beta-2 glycosidic linkage, and that's how glucose and fructose come together to form sucrose. Then up here I just have other forms of different fibers. So if you are into supplements, you may have heard of psyllium husk. You can buy psyllium husk powder online or at health food places. And we see here that this is just sort of a complex branching version that contains these arabinose xylans and these xylose and arabinose sugars, which are all just different epimers of glucose. And they form into these complex structures that have all these different sort of weird glycosidic linkages on them. And humans do not have enzymes to break down these sort of weird glycosidic linkages that happen on carbons, such as 2 and yeah, so 2 and the 3 carbon there. We do not have enzymes that can break those sorts of linkages. So fiber is uh, is is indigestible to humans, and so that's why. So there is energy in these bonds. So there is calories there are calories in these bonds. There's energy in these bonds, but since humans do not break them down, we actually do not get calories from this. And so this, for instance, psyllium remains undigested inside humans, and so this can be used as sort of a bulking agent that can sort of it in water, it sort of bulks up and forms sort of a slime that makes it so that we digest things slower, which can do things like, and we'll get into this a lot in later videos when we get into digestion. So this can do things like reduce glycemic index, and it can sort of collect other molecules into the bulk, which can then sort of move out and be excreted. So this is another type of fiber called xanthan gum. If you look at nutrition or the ingredients on the back of on the nutrition facts of different foods, you might see xanthan gum listed on it. And we see that this forms these weird glycosidic linkages. For instance, on the 3rd carbon here, we can have this repeating going on here. We see that we also, on our 6th carbon, have added this group here, which is not something you see in normal sugars. So this is called an acetyl group here. Actually, it's acetyl group. And we can have different substitutions here and here on these. So anyway, this is another agent that's often added to foods to give it different, essentially different textures. So this is added, I think it's added to things like doughs, like bread doughs, to give them a certain texture and like a certain consistency. Up here, I have Inulin, which is another supplement you may have seen at your health food stores or on places like Amazon, which is just another sort of fiber. So you see on the 4th carbon here, we're actually bonding to 2 different things, which is another it's another glycosidic linkage that humans cannot digest. Bacteria in our gut can actually digest this, which is usually what inulin is used for. It's a prebiotic, and so we do not gain calories from this, but our gut flora actually gains energy from this, and so that's why it can help the growth of gut bacteria. So that's what inulin is for. Psyllium husk is sort of a fiber in the traditional sense, in that it's indigestible, and that it sort of just moves through you and takes other things with it. So that's it helps with things like bowel movements and things like that, where prebiotic is supposed to help feed gut bacteria. Down here I show a different sort of fiber, and this is one we'll get into in a lot more detail in later videos. It's called glycogen, and this is the way that humans actually store sugar primarily in the liver. And so when we eat too much sugar in our meal, our body will actually uh, combine protein, or not protein, but sugar into this sort of large branching complex that has these alpha 1,4 glycosidic linkages and alpha 1,6 glycosidic linkages. And then this is, as you may remember, is very similar to starch, which humans do have enzymes to break it down. So in the liver, we can store it like this, but then if we are in a fasting state, we can actually break it back down into, so we can break our glycogen back down into glucose and release that into the blood for our other organs to use for energy. And obviously, this, if it sort of sits around too long, which might happen if we're just constantly eating sugar, glucose or glycogen can be broken down into glucose instead of released in the blood. And we'll go through this in more painstaking detail in later videos. It can be turned into fat. That's why if we're constantly eating glucose, that can cause us to gain weight. Down here is where I'm going to talk about the aldose-ketose isomerization. So we see here that glucose and fructose, if you remember from the very beginning of this video, I said we're aldose-ketose isomers of each other. We see glucose has this aldehyde, and then fructose instead has this ketone here, and that occurs because we can form the aldo-keto isomer. That's changing where the double bond is, and we will end up with this this enolate intermediate. But down here I have the actual reaction mechanism, and so I don't remember if I had that here, but anyway, actually that you can see that better up here in this enolate. This hydrogen here is going to be more acidic than your normal hydrogen because it's adjacent to this double bond here. And you can see that over here because if we pull a hydrogen off here, which leaves this with this negative charge, this can actually come down here and its resonance stabilized through this double bond with the oxygen, so that makes this hydrogen adjacent to the double bonded oxygen more acidic, and so a base like this could come in and deprotonate that off of there, giving us this double bond, and then this will pull off a hydrogen from water, which will give us this enediol intermediate. This hydride, or not hydride, this hydroxyl here can then pull a hydrogen off of this oxygen. We get the electrons collapsing down, and then this, instead of collapsing up here, will then pull a hydrogen off of water. We recapitulate this. This is acting as a catalyst, and the definition of a catalyst is that it is something used in the reaction but not used up. So we start with this and we end with it, and now our carbonyl has gone from this to this, and so this becomes our anomeric carbon, and that's why we see here in this sucrose that our anomeric carbon on here was on the 2 carbon. We had this sticking off here because this was formed from a ketone. If we go back up to our ring formation here, I said that this hydroxyl here on the 5th carbon is attacking the aldehyde carbon here. If this was fructose, then it would actually be attacking, we would actually have our double bond on this carbon here. So this would be attacking that. We would have this up here as a hydroxyl group. And so that would give us this down here, which has the ketone here. And that's why we end up with, in fructose, this 5-member ring with these two, the 6 carbon and the 1 carbon sticking off, with the anomeric carbon here at this this 2nd carbon. And that's what I have pointed out up here, is this this hemiacetal and hemiketal. So if we are using an alcohol, and this would be happening inside the ring in our sugar, if we have an alcohol attacking an aldehyde, then it forms this hemiacetal, which is this oxygen carbon to hydroxyl. If it's with a ketone, which we find in our fructose, then it forms this hemiketal, which yeah, instead of the hydrogen being here, we now have this here, and so our oxygen is bound to something that has 2 carbon species, or 2 carbons coming off of that carbon there, which is what we see in our fructose ring structure. We have this oxygen here bound to this carbon, which is then bound to this hydroxyl, but then we go to these 2 different carbon atoms here, and that's how our fructose is formed, from these Aldose-Ketose isomers of glucose, of which fructose is the Aldose-Ketose isomer of glucose, through this mechanism down here. This is another reaction that you can have occur with sucrose, which is caramelization. So if you've ever caramelized sugar, you put your sugar in a pan and you just add heat. So we're adding heat, and we end up having just all kinds of different complex reactions going on. So one of them is just going to be separating the glucose and fructose, but then as you continue adding heat, you start generating these species. So the maltol, which gives it the caramel taste, and the furan. And you see here we have these double bonds going on, particularly this, which has the alternating double bonds, which are called conjugated double bonds. And we'll run into this more in future videos. Conjugated double bonds, which has a large effect on the color of substances. And so this gives it a nutty flavor, but it also changes it from being the white color that you see in your sugars to having that brown color of caramel. And so it's a complex reaction. There's a bunch of different species. This isn't even all the different species of substances that are present in your caramel. And you'll still have glucose and fructose present. You'll probably still have sucrose present. Not all of it's going to be decomposed. And so caramelization is this complex reaction. And so there is also something called the Maillard reaction, which is if we do caramelization with sugar in the presence of protein, which is composed of amino acids. I'll just say AA there. And so that's just another complex, a complex sort of reaction that ends up having a bunch of sugar amino acid bonds together, and that's something that you see happening more often if you brown things like vegetables, like if you do it to onion or when you do it to meats. So there are sugars and proteins present, and so you have this Maillard reaction, which ends up forming. So when you brown your meat, that's what's happening is this Maillard reaction. And so over here, I just have different fake sugars. So this is Sucralose. This was erythrose, I think it's called. So these are just different fake sugars that you will see in your in your ingredients on the nutrition label of different foods. So fake sugars are things that taste sweet. So they taste sweet, but they are not indigestible. So they're not digestible by humans. So things like sucralose. So this, when it gets into the digestive tract, is so actually all these are not digested. I think some of these, I think this might be digestible by bacteria, but I'm not exactly sure. You might want to look that up to confirm, but I believe sucralose is not digested by anything. You can see here it has these chlorine atoms on it, and right here. So what can end up happening is this can actually cause osmotic pressure in your large intestine. So it sort of sucks water out of your blood into your large intestine, and that's why if you eat too much of this artificial sweetener, it can actually cause diarrhea because you are adding a lot of liquid into your stool. So you don't want to eat too much sucralose because it can cause this diarrhea. Over here we have aspartame, and we have it broken down here into its constituents, which is phenylalanine and aspartic acid, which are two different types of amino acid. And then we have this methanol added onto the carboxyl group of phenylalanine. And if you look at pops or sodas, if you're not from Michigan like me, you'll oftentimes see on the back of Diet Pop, it'll say, it'll have a warning for phenol, I think it's phenylketonurics, that it contains phenylalanine. And that's because in the aspartame we have this phenylalanine, this amino acid phenylalanine. And fetal ketonuria is just a disease where you don't properly digest phenylalanine, and so they want to warn the fetal ketonurics, people with phenylketonuria, that it contains phenylalanine. Um, and so yeah, this aspartame just contains aspartic acid, phenylalanine, and then it has this methanol group added onto the carboxyl of the phenylalanine. I'm not going to dwell on it too much, but I just wanted to touch on quickly uh, how sugar is made, and that I think most people have learned that it's from photosynthesis, which takes carbon dioxide out of the air, water taken up from the roots, light from the sun, and then it forms sugar and releases oxygen. So we see over here that this can be further broken up into what are called the light reactions, and then the Kelvin cycle, which you will also see as the light independent reactions, independent reactions, and so the light reactions are sort of where a lot of energy is generated, such that the energy intensive, the energy using process in the light independent Kelvin cycle can be used to generate sugar. And so the light reactions, so this it's kind of hard to read here, but this is energy. This is energy here. So light in the form of a photon comes in, it's collected, it excites an electron up in the energy here, and then by sort of through controlled methods moving the electron down the energy gradient, you are producing energy from this, and then light can hit another photosystem to gain energy into the electron here, and then once again moving the electron down the energy gradient to generate energy for the production of energy carrying molecules. And so we see here, this is the what we have here is the proteins that are involved in this process. So light hits the photosystem 2, and this is always a little bit confusing because light is the photosystem 2 is happening first over photosystem 1. I think these were named in the order they were discovered, not in the order that that this actually occurs. And so what you're actually doing here is using the energy from this electron going down the energy gradient, which is releasing energy for use. That's that delta G energy. And what that's actually doing is pumping these hydrogen ions, this acid, from the chloroplast stroma into the thylakoid lumen, which is just an organelle that's separated by a membrane, and so you can pump hydrogens up the gradient. That's why it needs energy, because the delta H wants diffusion to, or actually that's the delta S rather, wants diffusion to happen. So it wants the hydrogen to be equal on both sides. You use energy to pump these up the gradient so that we have a higher concentration here. And then you can use this ATP synthase. The hydrogen moves through it, and that allows ADP. This moving down the gradient. So if you remember, our delta G was our delta H minus delta ST. So this is using this delta S here to give us delta G by allowing these to diffuse back through this protein. It's generating energy and allows us to form ATP, which is our sort of energy molecule. It's sort of the $20 bill of energy molecules. This is sort of what's used as energy, and this a similar process is used in cellular respiration, which I will get to in painstaking detail in a later video. I'm just going through this quick right now, uh, uh, using this hydrogen gradient to produce ATP. Um, and you see here we're also producing what's called NADPH, which is just an electron carrier. Um, and so this ATP and this NADPH is then used in the light independent reactions, which is the Kelvin cycle. Uh, I liked this diagram here because uh, it shows that there's three molecules of this ribulose bisphosphate, and then we fix carbon dioxide to it to generate six molecules of this three phospho. I think that's phosphoglycerate. Yeah, and so that's the carbon fixation. Then you can see we use our ATP and our NADPH here to produce the six molecules of glyceraldehyde 3-phosphate, which this can then go to be turned into to glucose. But some of these five molecules of this remain in the cycle. Only a single molecule of it is shunted aside for the production of glucose. Then we use more ATP to generate this ribulose bisphosphate to turn five molecules of this GA3P into three molecules of this. Then we can affix three more carbons. This is just the same thing, but this actually lists some of the enzymes being used in this. So we see here the carbon fixation is using this ribulose bisphosphate carboxylase, and oftentimes you'll see it with oxygenase added onto it, which is why it's often shortened to RuBisCO, because this is the carboxylase and oxygenase. The oxygenase is not the point of this molecule. In fact, that's a side reaction that is not desirable. So the carboxylase, which is adding the CO2 onto the ribulose 1,5 bisphosphate to generate the 3-phosphoglycerate. And then we see here in the phosphoglycerate kinase, which is what's adding the ATP, or using the ATP to add phosphates. We have the glyceraldehyde 3-phosphate dehydrogenase, which is what's using the NADPH. We see down here the isomerase, aldolase, and fructose 1,6 bisphosphatase enzymes, which are part of glycolysis, which we will get into in painstaking detail in later videos when we start getting into metabolism. And so this is just, it can use molecules in this molecule here generated in the Kelvin cycle is an intermediate in glycolysis, and so that can be used in glycolysis for the biosynthesis of of, uh, of carbohydrates. And then we see here that we're going through, and then phosphofructokinase is the enzyme that's using this ATP here. This still shows, just kind of skips over a bunch of enzymatic steps there. I had a hard time finding a diagram that had any of the actual enzyme names in it. Not that I guess it matters that much. I'm not going to go through the enzymatic reactions in detail here. It's not that important for nutrition and metabolism, but I just wanted to quickly go over how carbohydrates are produced in plants. So I talked about this ribulose bisphosphate carboxylase, also with oxygenase activity, and that is this carbon fixation step. So Rubisco is the most abundant protein in the world. So every plant has Rubisco, and it has a lot of it because it's not a very efficient enzyme, and so it needs to make up for that by just having a ton of it. And so it's the most abundant protein in the world. And that is the protein that fixes carbon dioxide to sugar in this step up here. So we have our RUBP, that's this right here, and it adds the carbon dioxide while also splitting the molecule. So we see our carbon dioxide has been added right there, and it turns it into 2 molecules of 3-phosphoglycerate. And this is the mechanism here. So we're still using that hydrogen adjacent to the double bonded oxygen, which will be more acidic, so that will have a lower pKa than, say, this hydrogen here, because of that resonance stabilization that we see here in the enediol. And then in the enzyme, we will actually have these electrons collapse down, and the electrons in this double bond will actually be used to bond to the carbon dioxide here, which then pushes those electrons up to that carbon. And so we end up with the carbon here from our carbon dioxide, and we've recapitulated this oxygen double bond right here. But then we have water come in and act as a nucleophile on this carbon, which has the so this has the partial negative because this double bonded to this oxygen. This has the partial positive, so that will attack this carbon here, pushing those electrons there. And because this is in an enzyme, this will be attacking this carbon. So I talked about before, we have the carbon R1, R2 double bond, and it can attack from either the back side or the front side. And the enzyme will steer it so that it always attacks that carbon from the same side. There will be the active site of the enzyme will be such that water can only access this carbon from the same side, so that it controls the chirality, the stereochemistry of this carbon. And so the water attacks that. We end up with these electrons collapse down, which then allows this bond here to break, and that's how we end up with 2 molecules. So so that is the reaction mechanism of Rubisco, which is kind of this important part of taking the carbon dioxide out of the air, and that's how it's turning carbon dioxide into sugar. And down here, I said before that we can have this carboxylase activity, which is this. This is what is desired in Rubisco. We want the C part, but it can also fix uh, oxygen, which has this undesirable effect. And plants uh, actually have evolved a pathway that can sort of turn this back into something that's useful, that can then be turned back into something like this. But it uses energy, so it's an energy, sort of an energy wasting sort of thing going on. And so plants have evolved ways of trying to keep oxygen out of the areas where Rubisco is performing its function. But in case this happens, it also has this. It's actually a kind of complicated pathway. I'm not going to go into it. To turn it back into something useful. So this is an undesirable effect. Plants want this carboxylase activity happening. That's sort of the business end of this. This oxygenase activity is undesirable. And so that's just a quick overview of photosynthesis. I don't want to go too much into it. Yeah, and so that's what I wanted to cover. Just sort of talking about carbohydrates before we discuss it in terms of nutrition and metabolism that that takes place in humans. So human metabolism. All this stuff is stuff that is sort of independent of human metabolism. It's stuff that's exploited and utilized in human metabolism. But this, what I talked about before, was just looking at carbohydrates from a purely organic chemistry standpoint. Then I went into the biosynthesis of carbohydrates, which happens in plants, not in humans. And so that's basically just me. I wanted to introduce carbohydrates as sort of a as so carbs as sort of a player, sort of a player in our that will that's misspelled player that will be that we will be running into a lot as we get into metabolism in future videos. In the next few videos, I'm going to discuss I first discussed carbs. The next player I'm going to discuss is lipids, and I will talk about that in a purely organic chemistry, in terms of organic chemistry. After that, I'm going to talk about amino acids, and I'll talk about that in purely organic chemistry manner. Then I'm going to talk about proteins, and that will be in a somewhat more uh, a chemistry point of view, a structural biochemistry point of view. In fact, I will be talking about the structure of proteins, which are are peptides, are polymers of amino acids, which are called peptides. And so that's what the next few videos will be, is talking about these biologically interesting molecules from a sort of organic chemistry point of view. And then I will talk about proteins, which are obviously interesting as far as a source of nutrition goes, but I will be discussing it more in a structural biochemistry point of view because that will be how we understand how we understand things like enzymes and receptors and things like that in future videos. So anyway, I've talked long enough in this video. So that is our first key player in nutrition and metabolism, carbohydrates. And I will see you in the next video.